going to talk about cell transport. As we go through this, make sure that you guys are going through the self-paced Nearpod, answering those questions, because that will be your grade for today. On top of the case study. So we're going to talk about the cell membrane first before we talk about cell transport. So the plasma membrane or the cell membrane means the same thing. They form a boundary between a cell and the environment outside. And it controls the passage of materials into and out of the cell. So we see here there's a lot more than just a little wall between the outside and the inside. There's a lot that goes on here, whether it be through cholesterol, channel proteins, phospholipid bilayer, carbohydrate chains, glycoproteins, other proteins that are here as well. So the first thing we're going to talk about is cholesterol. So the cell membrane is made up of a double layer of phospholipids, and they interspread within a variety of other molecules, and one of those is what we call cholesterol. And with cholesterol, it's kind of like a love-hate relationship with the cell membrane, because they're going to embed themselves into the hydrophobic portion of the lipid bilayer to stabilize the membrane, to make it less permeable, but it also makes it more stable so that the cell membrane can be held together through some force as well. So I mentioned that embeds itself in the hydrophobic part of the phospholipid bilayer. So what is a phospholipid bilayer? And as a molecule composed of three basic parts, we have a charged phosphate group, we have a glycerol, and two fatty acid chains. And so a great way to remember what a phospholipid bilayer is this. So a lipid looks something like this. So this is my glycerol. These are my fatty acid chains, which you see in the green. So these are fatty acid chains. If I can draw my little dot here. FC for fatty acid chains. There's three of them. So that is a lipid. And so what is actually occurring here is we're going to get a phosphate group added. Here, we're going to lose a fatty acid chain. So this was a phosphate lipid, or in this case, a glycerol and fatty acid chains. Now, because it's a phospholipid bilayer, it has two layers. So as you can see in this picture, we have layer number 1, layer number 2, and they are arranged where the phosphate heads are facing the outside space or the extracellular space or into the cell, and then these two fatty acid chains are inside. And so the charged head is what we call hydrophilic. It loves water. Philic, love, water, hydro. So it loves water. It's polar, while the fatty acid tails are uncharged, and the tails are nonpolar. They're hydrophobic or water-fearing. Hydro-water-phobic fearing. So the lipid bilayer forms the cell membrane. The hydrophilic heads, as we talked about, are on the outer layers, while the hydrophobic tails form the inner layer or away from the water. So the cell membrane also has a number of specific proteins embedded in the bilayer. We've kind of talked about this as we've gone through the year. But we have two different types of proteins. We have a transmembrane protein, which literally goes to the entire protein, while the peripheral protein is associated to one side or the other of the bilayer. Now, the cell membrane has been described as a fluid mosaic model, and this is due to the fact that proteins are able to move around in the bilayer. And so as we talked about, there are sometimes that we need certain organelles or certain materials on certain sides of the cell at that point. So these membranes are allowed to move this way or this way but they cannot flip vertically, so it can't go from this way to all of a sudden start pointing to the other side of the protein. Or these transmural membranes aren't allowed to flip. So a cell membrane has the property of being semi-permeable, or what we call selectively permeable. So semi-permeable meaning molecules can cross without the aid of protein, but also can be selectively permeable, where non-lipid soluble molecules can pass the membrane with the use of transport. So it's semi-permeable, where certain things can go through, while it's selectively permeable, where our proteins can transport things across as long as it's a certain thing or a certain molecule. So you see the picture to the right, some things that can go across without the use of proteins. So receptors are proteins which detect the signal molecules perform an action in response, or what we looked at with enzymes, right? And so these are recognized and bind certain specific molecules known as ligands, or what we called earlier reactants or substrates. Binding to a ligand causes the receptor to change its shape or conformation to allow it to perform a function, in this case, an enzyme. So intracellular receptors are found within the cell. These molecules must cross the membrane in order to bind to the receptor, and these ligands usually are nonpolar and relatively small, such as hormones produced from the smoothie R. And you can see here, hormone comes in, binds to receptor goes in to perform a certain function. In this case, maybe mix something to break down sugar or something like that. Now, membrane receptors bind to ligands outside the cell when they produce a cell to produce an action within the cell. These molecules inside perform a complex cascade reaction to allowing the cell to, in response to the ligand binding. And so this could be dealing with, in this case, epinephrine, for body response. We can also deal with this as well with bacteria, and we have white blood cells going through a cascade. Now we're going to be able to identify molecules in a plastic membrane that provide basic membrane structure, cell identity, and membrane fluidity. So now we're going to talk about cell transport. So 2 questions you'll be able to answer are: what are the processes of diffusion, facilitated diffusion, active transport? What is the effect on hypotonic, hypertonic, and isotonic solutions in a cell? So as we were talking about, we have the lipid bilayer, right? So we have the hydrophobic heads and the hydrophilic heads and the hydrophobic tails. And so cells must continually import and export substances. So some molecules can enter and exit without requiring energy, and this is known as passive transport. And so a lot of the times you'll see as a high concentration outside the cell to a low concentration inside the cell, like we did look with water. So molecules are going to move from a high to low. And if we see a high to low, we're going to think passive transport if there's no protein there. So diffusion is the movement of molecules in a fluid or gas from reaching a high concentration to a low concentration, like we looked at osmosis with water. So it is a result of natural motion of particles, and they result in equilibrium. So we have a concentration gradient. So a concentration gradient is the difference in concentration from one location to another, and molecules will diffuse down their concentration gradient from a high to a low concentration. So a way to kind of go through this is, if you have, you guys probably remember like the Axe spray. Well, if someone sprayed Axe in the edge of a classroom or in the hallway or something, you could probably smell it a little bit later. Well, depending on how far you are will determine how much of it you smell, and also how long it would take for you to go from it being sprayed to you smelling the axe. So many factors affect the rate of diffusion. We have temperature. Higher temperature diffuses faster. Surface area, a larger surface diffuses faster. Concentration gradient, high gradient diffuses faster. Smaller particles will diffuse faster. And then, of course, in gas, molecules are moving the fastest, so therefore it's going to diffuse faster. Now, one of the things with surface area, as we looked at with cell respiration, was the cristae in the mitochondria. It increases the surface area so we can bring oxygen into the mitochondria so that, or more oxygen, so we can produce more ATP in a larger surface area. So now, diffusion can only occur if the cell membrane is permeable to the substance and there is a concentration gradient from one side to the other. And so one of those things is, you guys probably saw this on the interactivity that we did. So you saw this right here. Our tube. There was a selectively permeable membrane. We had a lot of an increase of glucose here. We had a decrease of glucose here. Or sugar, and in the directions it said that only the membrane was selectively permeable to water, so, or that glucose could not pass through. And so as we can see here, we have an increase in glucose, a decrease in glucose here. And so as you guys saw in the POGIL, we got to try to reach equilibrium of that ratio of water to glucose. So water will move in this direction. It's the same thing as you see here to the right. Except in this case, water is going to move more to the left. Water's going to move to the left. In this case, this solute is permeable. It's going to move to the right. But if not, then water would move to the left. This water would increase in size so that we have an equal ratio. So, we already talked about this, but again, it's very, very important. Okay, cell membrane has a property of being semi-permeable, meaning that smaller molecules can cross without the aid of proteins, and it's also selectively permeable because they can have the help of proteins of selective material to go through or out. So in cells, non-polar substances and smaller molecules can diffuse across the membrane, so like our carbon dioxide and oxygen. This is kind of a review from what we talked about on the POGIL. But osmosis is the movement of water from a high area of concentration to a low area of concentration. And it is important to recognize the higher concentration of dissolved particles in solution and the lower concentration of water. So as we see here, this was that gated channel we looked at previously. Here's what it would look like if just water is moving. So, kind of review back from the POGIL, we have hypertonic, isotonic, and hypotonic. So hypotonic solutions have less solute and more solvent. So it has more water outside and more of something else, which is the solute inside the cell. And so water is going to go into the cell. And as I like to call it, hypo, big as a hippo. On isotonic solutions have the same amount of solute and solvent inside and outside the cell. And then hypertonic solutions, they have more solute and less solvent outside the cell, so water's going to rush out, cell's going to shrivel up. So here we go, here's the direction of osmosis, here's that gated channel again, and so we can see semi-permeable membrane, as we can see what hypertonic and hypertonic do, and where the water's going to move based on each scenario. So it's going to go from hypo to hyper to equal out, so we have more water in hypo, and then in the isotonic, water is going to move back and forth equally, and then hypertonic, it's going to move to hypertonic because it has more solute. So under normal conditions, the osmotic pressure in the fluids in your body and your cells is isotonic, like this red blood cell here. However, if you put the cell in a hypertonic or hypoosmotic solution, then the water is going to travel outside the cell into the solution because you have more salt inside the cell. While if a cell is put into hypotonic or hyperosmotic solution, then water is going to travel into the cell from the solution leading into systolis or cell death or lysis. So some molecules cannot easily diffuse across the membrane. They require the use of membrane proteins to facilitate the movement. So this is what's going to be called facilitated diffusion. So this allows molecules that are not lipid soluble to pass through the membrane through the use of channels. This is still going to go through the concentration gradient from a high concentration to a low concentration. And in this case, the number of carrier molecules is also a limiting factor. Carrier proteins serve to transport molecules down their concentration gradient. An example of facilitated diffusion is that glucose is in a cell, and this would be through facilitated diffusion. So we see a high number of glucose outside the cell. We're trying to get glucose in the cell so we can create ATP, or use that energy, and so we're going to use the help of these carrier proteins through facilitated diffusion. We also have ion channels, and they transport ions down the concentration gradients, and each type of ion is specific for the ion it transports. Some ions are gated, meaning they need to open to allow transport of ions. So we see here we have potassium as its ion-specific channel here. And so there are three ways that gated ion channels can open: through voltage, electricity, a ligand, where we have a substrate attached to it for it to open, and also mechanical. So in many cases, cells must move materials from a low concentration to a high concentration, or against the concentration gradient. And such movement is known as active transport. And unlike passive transport, active transport requires energy or ATP. So looking here real quick, let me go back here. Yeah. Okay. So we're going to go back to that picture in a second. So active transport moves particles through the membrane of the region to a low concentration and to a high concentration through a transport using energy. And up to about 40% of the cell's energy supply is used for active transport molecules to go against the concentration gradient. As we see to the left here. So before we talk about that, so I want to talk you through these three types of transports you'll need to know. And it might give you examples, it might give you a picture. These are going to be very, very important for you. So diffusion, right, or passive transport has two different types of diffusion. We have diffusion and facilitated diffusion. The big difference here is diffusion goes across the membrane with no help. Facilitated diffusion needs help, so we're going to have a membrane or a protein here to help it across. The biggest thing between facilitated diffusion and active transport, yes, they both need membranes. However, ATP needs to go in there, as well as we go from a low concentration to a high concentration in active transport. And so one of those examples is through the sodium-potassium pump. Now, many human cells have a high concentration of sodium ions outside the cell and potassium inside the cell. And so we need to create this concentration gradient for neurological communication, especially within contraction of muscles. So we can see here, here's a sodium-potassium pump. If you want to read through each one of these, you can. It just talks about how we have a concentration gradient and the step-by-step of it. Now, the sodium-potassium pump is required for maintaining homeostasis within the neurons of your body. So the biggest thing is contrast how facilitated diffusion is different from active transport, and you should be able to know that by now, and that will be a question that you answer on the Nearpod. And that's it. Hope you guys have a great day, and sorry I couldn't be with you, but I will see you soon.